So we are dealing with the paper 1 and chapter 2, that is the cryptogams, and in that we are studying the pteridophytes. Okay. Now as we have studied the structure and as well as the reproduction in the pteridophytes, now we are going to consider some very special and the very important topic from the UPSC mains point of view, like the origin and evolution of the pteridophytes. Okay, stellar evolution, homospory, heterospory, all these topics are very much important from the UPSC mains examination point of view. So for today's lecture, we are going to focus on origin and the evolution in pteridophytes. Okay, how the pteridophytes got originated, different theories and different hypotheses are there to explain these things, and how these pteridophytes got evolved. Okay, this is the topic of today's lecture. So origin and evolution in the pteridophytes is a very important lecture, and you should focus on this particular lecture very carefully, because there are lots of chances that UPSC may ask you this question for 20 or even for the 5 or even for the 20 marks. Okay, so consider this is your syllabus, that is the paper 1 chapter 2 cryptogams. We are here and we have to study the structure and the reproduction from the evolutionary viewpoint, and at last we have to study the ecological and economic importance of pteridophytes as well. All right, now consider the introductory slide for this. See, the pteridophytes evolved in the Silurian period and subsequently diversified in the Lower Devonian period. Okay, so it's a basic point, as you already know it from the 12th examination and from the 12th syllabus, that the pteridophytes were first originated in the Silurian period. Okay, they got evolved in the Silurian period and later they got diversified in the Lower Devonian period. Okay, so the Lower Devonian period is considered as a dominant period for these particular pteridophytes. Now there are various controversies regarding their origin and the evolution. Of course, there is no one view. Just like when we have considered the origin and the evolution of the Bryophytes, there are the many views, and finally we cannot conclude which theory is best to explain the origin of Bryophytes. In a very similar way, we are not sure which type of the evolution happened in the pteridophytes as well. So there are many theories and there are many controversies regarding the origin and the evolution of these particular pteridophytes. All right, now there are mainly two views regarding that. See, regarding the origin of the pteridophyte, there are mainly the two views. According to first view, the pteridophytic ancestry is to be sought from the Bryophytes. That is the Bryophytes where the plants from these pteridophytes were originated. Okay, this is the first view, and from your common sense, this is the obvious view, because as you know, from the on the evolutionary scale, there is algae, right? From algae there is a Bryophytes, and from Bryophytes there must have evolved the pteridophytes as well, right? So on the evolutionary scale, this looks with very common sense that the Bryophytes may be the ancestors for the pteridophytes, because they shows the much differentiation than the algae, and from the Bryophytes, the pteridophytes also shows the higher differentiation as compared to the algae and the Bryophytes. Okay, so the Bryophytes looks the very natural group as an ancestry for the pteridophytes. But is it true? Is there are any sufficient evidences to support this? Now let us consider, this is the first view, that the pteridophytes were originated from the Bryophyte, while according to the second view, the pteridophyte arose from the algae and evolved parallel with the Bryophyte. This is very important. See what they are saying: the pteridophytes were evolved from the algae. Okay, that is, the pteridophyte were directly evolved from the algae and they evolved parallelly with the Bryophytes. That literally means, what is the viewpoint of this hypothesis is that the algae giving rise to the Bryophytes as well as the pteridophytes on the parallel lines. Okay, that means the some algal organisms got converted into the Bryophytes and some of them got converted into the pteridophytes. That is, the parallel evolution occurred between the Bryophytes and the pteridophytes. That is, they co-evolved. Okay, they evolved together from the algae. This is the second viewpoint. So according to the first viewpoint, the pteridophytes on the evolutionary line straight away got created from the Bryophytes, but according to the second view, the algae is responsible for giving rise not only to the Bryophytes but also to the pteridophytes in a parallel evolution. That is, co-evolved, that is, pteridophytes and Bryophytes evolved together from the algae. Okay, this is the second viewpoint, and around these two viewpoints, the origin and evolution of pteridophytes rotate itself. Okay, so whenever we talk about the pteridophytes have main two views regarding their origin. First view say that they are coming from directly from the Bryophytes, and second view coming from the algal viewpoint, that is the algae, and algae is responsible for giving rise to the pteridophyte as well as the Bryophyte. Now let us elaborate this. First is the algal origin. Okay, now this concept of the algal origin is based on the similarity between the algae and especially the member of Chlorophyceae with that of the pteridophyte. See, what are the supportive things which are there for the algal origin? See, if you are saying that the pteridophytes were evolved from the algae, there must have been some evidences to support this. Now, evidences for this theory lies in the similarity between the members of the chlorophytes and the pteridophytes. Now, you will say that, sir, there are not much things which matches between the chlorophytes and the pteridophytes. Well, if you observe carefully, there are certain points which exactly similar between the members of the chlorophytes, that is the algae, and the members of pteridophytes. But the things like the vascular bundle, that is the xylem and phloem, vascular tissues, are typically absent in the chlorophytes, but they are present. They are very well developed in the pteridophytes as compared to the algae, right? So there are dissimilarities, but there are also the similarities. So the algal origin concept is dependent on the similarity between the algae, especially the members of the chlorophytes, with that of the pteridophytes. Now let us consider which type of the characters were matching between the chlorophytes and the pteridophytes. Okay, now consider, see, some of the common characteristic between them is the thalloid gametophyte. Of course, we have studied the pteridophytes, and in the alternation of generation in the pteridophytes, you will find the gametophyte which is the thalloid. Okay, there is a thallus like structure which forms the gametophyte in the pteridophytes. So in the alternation of generation, there is a sporophytic generation which looks like this, which is differentiated into the root, stem, and leaves. Right, leaf, root, stems are there in the differentiation of the body of sporophyte in the pteridophyte, but the gametophyte is the thalloid. Okay, only a thallus is there, and the antheridium and archegonium born on this particular thallus. Right, same is true for the algae as well. When you study the alternation of generation for the algae's gametophyte also shows the thallus, thalloid plant body in their gametophyte. Okay, so the gametophyte of the pteridophyte and the gametophyte of the algae both shows the thalloid gametophyte. Now, second, similar photosynthetic pigment, that is the chlorophyll A, chlorophyll B, and carotenoid A. In the pteridophyte, you will find the chlorophyll A, the chlorophyll B, and the other pigments like a carotenoid A. All these pigments are also present in the algae as well. Okay, so algae also possessing these pigments, and as you know, the algae, after the cyanobacteria, algae were the first typical plants to possess this photosynthetic pigment, different kinds of photosynthetic pigment, because as you know, in the cyanobacteria there is no presence of the chloroplast. Okay, only the chlorophyll is there and chloroplast is absent, but the algae possess the well developed chloroplast, and it contains the different types of pigments, and these are different types of pigments were also present in the pteridophytes as well. So from this you can say that the algae is quite responsible for passing out these pigments to the pteridophytes, because they show the remarkable similarity. Then both of the cell walls are made up of the cellulose. Okay, if you consider the cell wall of the algae and the cell wall of the pteridophytes, both are made up of the cellulose. Okay, that is cellulose, hemicellulose, and pectin is a typical chemical composition of both, that is the algal cell wall and the pteridophytic cell wall. Plus, they shows the starch as a reserve food. As you already know, in the last lecture we have seen this, they store the food material in the form of starch in the pteridophytes. So that the algae is also storing the food in the form of starch. Okay, so starch is as a food reserve present in the pteridophytes, and it is also present in the algae as well. So there are many similar common characteristics between the pteridophytes and the algae. First, we have studied that they both showing the thalloid gametophyte. Second, they show the similar photosynthetic pigment. Third, the cell wall composition is same, and they both show the starch as their food reserve. Now, see the fifth one, that is they both show the flagellated sperms. Okay, that is the male gamete. Okay, in the male gamete of the algae, they show the presence of flagella. In the male gamete of the pteridophyte, they also shows the presence of flagella. Sometimes multiflagellated sperms are there. So whether it's the algae or it's a pteridophyte, both are showing the flagellated gametes. That is the male gametes. Male gametes are flagellated in both. That is in the algae and the pteridophytes. Then water is absolutely essential for the algae in order to achieve the fertilization. Same is true for the pteridophytes as well. Okay, pteridophytes also requires water for their fertilization, so that the algae is also requires the water for their fertilization. Right? Plus, there is cell plate formation during the cytokinesis. Okay, during the process of cytokinesis, there is a formation of cell plate in both in the pteridophytes as well as in the algae. Okay, so as you already know, cytokinesis is the division of the cytoplasm, and during the division of the cytoplasm, the cell plate formation is there in the algae as well as in the pteridophytes. So, from these all 7 points, what you can conclude that there are many characteristics which are similar between the algae and the pteridophytes. Okay, so pteridophytes can literally match with the algae, and you can say that the algae can be the ancestry for these particular pteridophytes. Okay, so by studying all these points, you can say that the algae can be the ancestral material for the pteridophytes. Now, if we applied our common sense, because we have already studied the algae as a group and the pteridophyte as a morphological, anatomical, and reproductive characteristics, we have already studied it. So, from these points, from this earlier knowledge, you can conclude, you can also point out that there are many dissimilarities between the two. For example, the sporophyte of algae and the pteridophyte of the algae never matches with each other. Why? Because the sporophyte of the pteridophyte is differentiated into root, stem, and leaves. Such structures were not found in the algae. Now, if you consider the plant habit of the pteridophyte, which can live on the land, such characteristics are not possessed by the algae. Okay, differentiation in their body structure not found in the algae, but it is largely found in the pteridophytes. So same is true for the vascular bundle, like the xylem and phloem conducting tissue are typically absent in the algae, but they are present in the pteridophytes. So you can say that there may be some similar points, but there are also some dissimilar points as well. See, when we consider any hypothesis tends to take you to the to justify their points. Okay, how they are justifying their points, how the algal origin hypothesis justifying their point, but just by showing the similarities. Okay, they do not talk about the dissimilarities, but there are dissimilarities. Okay, of course we can judge it by the common sense, right? But from the algal origin, we have studied the 7 typical common characteristics between the algae and the pteridophytes, which are showing there may be the algal ancestry for the pteridophytes. Okay, now consider there are some authors which supported this hypothesis. See, one of them is the Lignier, and he has proposed another hypothesis which is supporting the algal origin. Okay, he supported the algal origin and he postulated that the pteridophytes arose from the chlorophyta, that is the green algae. Okay, from the green algae the pteridophytes got originated according to Lignier. Okay, what is the Lignier saying? According to Lignier, there is an algal ancestry of the pteridophytes, and particularly the chlorophyta, that is the green algae, that is the chlorophyce, is responsible for giving rise to the pteridophytes. Okay, with a dichotomizing parenchymatous thallus. Okay, because these chlorophyta possess the thalloid plant body, and there is a dichotomous branching, and these chlorophyta members were responsible for creating these pteridophytes. Okay, now see, for the transmigration from water to the land, the basal part entered the soil. See what he is saying, Lignier. According to Lignier, when this algae, that is the chlorophyce algae, when it move from water to the land, okay, the first requirement to move from water to the land, that they should have fixation in the soil. And for this fixation, the lower part of the thallus, this is the thallus which is found in the algae, and the lower portion of this thallus created some structures while transmigration from water to the soil, and it is used to fix itself into the ground or into the soil for the anchorage and absorption purpose. So when we talk about the Lignier hypothesis, what he is saying? He is saying that the pteridophytes are originated from the green algae. And these green algae, when they transmigrate right from water to the soil, they have developed the special structure from its lower thallus, which later we called it as a rhizoids. Okay, just like in the Rhynia. Okay, we are calling it as a rhizoids. Okay, which are developed from the lower part of the thallus. So the rhizoids in the Rhynia can be connected with that of the lower portions of the thallus in the algae. This is according to the Lignier. And according to his viewpoint, what he has imaginated is that the chlorophyce members of the thalloid body of the chlorophyce members has developed somehow the root-like structure which are called rhizoid to move from water to the land. That is transmigration from the water to the land. And this structure is specially created to anchor into the soil and to absorb water from the soil. Okay, so this is the Lignier hypothesis. All right. See, erect parts, on the other hand, the above portion part, that is the erect part, retained the photosynthetic function. See, observe, this is an Rhynia. Okay, and you consider that this is an rhizome and this is an aerial portion. Now according to Lignier, what happened? Consider this is an algae, okay, thalloid algae, and when this thalloid algae got transmigrate from water, consider this is in the water, and now it got transmigrated into the land. Okay, now when it got transmigrated to the land, it required the special structure to anchor to the soil. Okay, so he developed the rhizoid from the lower parts, while the upper portion, which is the photosynthetic portion in the algae as well, this upper portion, this erect part retained the photosynthetic function and it got erected like this. Okay, and it retains the photosynthetic function of the thallus. And the aerial portion with the terminal sporangia become the primitive three-dimensional dichotomous branching system. For example, the rhinia. Okay, see, in the rhinia, what you will see, the trichotonomous branching is there. Okay, that is the dichotomous branching is got converted into the trichotonomous branching. Three-dimensional branching is there, and this three-dimensional branching later bears the sporangia. So, according to the Lignier, what he is saying, the lower portion of the thallus of the algae got converted into the rhizoid, and the upper portion is got converted into the erect part, which is showing the photosynthetic function, as well as the apex there is a presence of sporangia. Okay, so Lignier trying very hard to support the algal origin, and he is saying simply from the morphological characteristic that the thallus of the algae from its lower portion give rise to the rhizoids, and from upper portion it has given rise to the erect portion of the pteridophytic plants, that is, for example, rhinia. Okay, so according to the Lignier, the morphological transformation occurred in the algae to create the pteridophytes. All right, now consider the Church hypothesis. Okay, of course, Church is also believing that the algal origin is there for the pteridophytes. See, the Church is a believer of the polyphyletic origin of the pteridophytes. That literally means the algae, the many groups of the algae has given rise to the many groups of the pteridophytes. Like this, okay, polyphyletic origin, not the one phyla, not from the chlorophysi, but all the members of the algae are responsible for giving rise to the present day pteridophytes. Now, when we talk about the pteridophytes, see, when we talk about the pteridophytes, pteridophyte is a diversified group, okay, because as you know, while studying the classification, we have rightfully described the classify the pteridophytes into the 5 categories. Okay, so all these 5 categories were descended from the different algal groups. This is according to the Church. Okay, what a Church is saying, there is a polyphyletic origin for the pteridophytes from the algae. Okay, and he proposed the theory as a Thalassiophyta and the subaerial transmigration. This is the title of his theory. That is, there were the hypothetical group which is called as the Thalassiophyta, and there is a subaerial transmigration. See, let's elaborate this. See, according to him, the hypothetical group of the advanced marine seaweeds, which is called as the Thalassiophyta, okay, it's a hypothetical group. That literally means he has imagined this group, okay, because at present this group is not there, and even the fossil records were not found for this particular groups. Okay, so there is a hypothetical group which of the advanced marine seaweeds called as the Thalassiophyta. This Thalassiophyta is the group of the marine seaweeds, okay, and it is from the ancestral stock for the land plants, that is the Bryophytes and the Pteridophytes. So there is an imaginary group which is called as the Thalassiophyta, and this Thalassiophyta act as a reserve group for the creation of the Bryophytes and the Pteridophytes. That is, it is an ancestral stock for the creation of Bryophytes as well as the Pteridophytes. Okay, now see, the Church hypothesis, it is very difficult to understand. Why? Because he is considering some hypothetical group, that like the Thalassiophyta. Now, how the Thalassiophyta looks like? Now, he has considered all the characteristics, that is the algae, Bryophytes and the Pteridophytes, to be mixed with the Thalassiophyta. That is, there is a one group which is showing the characteristics of the algae plus the Bryophytes plus the Pteridophytes. Okay, and this group later give rise to the Bryophytes and the Pteridophytes. This is according to the Church hypothesis. Now, see, this transmigrant algae had a metabolic efficiency of the chlorophysi. Like the chlorophysi, like metabolic efficiency were present in this Thalassiophyta, and somatic equipment and the reproductive scheme of the Phaeophysi. That is, the reproductive ways of this Thalassiophyta belongs to the Phaeophysi, while the metabolic machinery corresponds to the chlorophysi. That is why he has rightfully explained it as a polyphyletic origin, because not only the chlorophysi but the Phaeophysi characteristics were also found in these Thalassiophytas. So, Phaeophysi, some of the somatic characteristics and the reproductive characteristics of the Phaeophysi were present in the Thalassiophyta, and some of the metabolic machinery, whatever the metabolic efficiency is there, it belongs to the chlorophysi. So, Thalassiophyta is nothing but the combination of Phaeophysi, chlorophysi, and rest of the Bryophytic and Pteridophytic characteristics. And from this Thalassiophyta, the present day Bryophytes and Pteridophytes were evolved. This is according to the Church. Okay. Now, Church is strongly advocating that the Pteridophytes and the Bryophytes were evolved from the algae, because he has created an hypothetical group which is called as the Thalassiophyta, and this Thalassiophyta is nothing but the combination of algae, Bryophyte, Pteridophyte. In that particular category, the chlorophysi, Phaeophysi characteristics were assembled in this particular Thalassiophyta, and these Thalassiophyta later give rise to the Bryophytes and the Pteridophytes. This is according to the Church, okay, and this is called as the Church hypothesis, which is strongly supporting the algal origin for the pteridophytes. Okay, now see the Fritsch hypothesis. Okay, what Fritsch has to say about this algal origin. He suggested that the green algae ancestor gave rise to the both bryophytes and pteridophytes. Okay, of course he is also the supporter of the algal origin, and he is saying the same thing, that the bryophytes and pteridophytes were came from the green algae. Okay, now see the green algae, he elaborated this. See, the green algae is the heterocytus in habit. Now what is meant by heterotrichous? Heterotrichous literally means the one we showing the prostrate habitat as well as the erect habitat. Okay, so prostrate, if there is a presence of the prostrate as well as the upright system, then we say that it is an heterotrichous habit. You know, when we study the chlorophytes, there are some members which are showing the erect habit, and there are some members which are showing the completely uh, prostrate habit, right? So there is a prostrate habit, there is a erect habit, and that is why we are describing them as a heterotrichous. Okay, so this is very important. This is important observation by the Fritsch, because if you consider with the common sense, the aerial portion and the basal, these thallus, which are prostrate thallus, these are considered as a basal portion of the pteridophytes. Observe, if in the chlorophytes group we have the prostrate algae, and in the same group we have the erect algae. See, if we combine these two habits, what it looks like? It looks like the Rhynia plant. There is a thallus, which is a prostrate thallus, this rhizoid portion, and there is a erect branching. So, according to Fritsch, he has made the important observation, in the chlorophytes, what you will find the prostrate habitat as well as the upright system. So by combining the prostrate habitat and the upright system, there is a creation of the bryophytes plus the pteridophytes from the ancestral stock of the green alga. Okay, and he is rightfully given the morphological evidence that the chlorophytes member shows the erect as well as the prostrate habitat, and when we combine both these habitat, you can imagine a pteridophyte, because it also showing the prostrate rhizome and the upright branches. Upright axis is there, upright stem is there, and there is a horizontal rhizome. Okay, so when we consider the heterotrichous habit of the algae, we came to know there were two types of habits shown by these chlorophytes members, and when we combine these two types of habit, it will create the bryophytes or pteridophytes, right? See, the apical growing point was established in the upright branches. What is saying? What are the upright branching is there in the algae? At the apical region here, the growing point were established in the evolution, and thus the heterotrichous thallus give rise to the erect land plants, just like this, and the growing point were developed there, and that is why it has given the erect habitat like this. Branching, erect branching is subaerial branching is like this, is created out of this apical growing point. Okay, thus giving rise to the erect land plants by elaborating the erect portion and by diminishing the prostrate part. Okay, so prostrate part is got diminished over a period of time, and this growing point has given rise to the aerial structures like this. Okay, but still in the fossilized plant like Rhynia, we find the prostrate as well as the erect habitat, which is suggesting that the Fritsch hypothesis has some weightage, because these morphological characteristics are strongly matches with the chlorophyte habit, which showing the prostrate as well as the erect habit. Okay, so prostrate and erect habit, when we combine them, it looks like the Rhynia plant, and according to the Fritsch, apical growing point were established in these particular upright branches of the algae, and these due to this apical growing point, it has created the erect land plants. Okay, and by elaborating the erect portion and diminishing the prostrate part, so in a meantime, the lower portion is got diminished and it got converted into the roots only, and that is why today's pteridophytes, that is the which are differentiated into the root, stem, and leaves were got created out of these particular algae. Okay, so this is how the pteridophytes or the bryophytes were evolved from the green alga. This is according to the Fritsch hypothesis. Okay, so Fritsch is also supporting the algal origin. All right. Now consider the Andrews hypothesis. He is also suggesting that the pteridophytes or the bryophytes were evolved from the algae. See, based on the fossils of the marine algae such as the nematothallus, then the protosalvinia, then the Phyton, the Andrews hypothesized that these algal groups were independently give rise to the different groups of the earlier vascular plants, that is the pteridophytes. So what is Andrews hypothesis? Andrews hypothesis suggests there are the fossil record which are found for the marine algae in the they were named as the nematothallus, then the protosalvinia and the Phyton. These groups were independently give rise to the present day vascular plants. Okay, these present day vascular plants are there just because of the marine algae which are now extinct. Okay, it is in a fossilized form. Okay, and when we study these fossilized form, their morphological characters matches with the early vascular plant, and that is why Andrews hypothesized that these earlier vascular plants may have evolved from these marine algae fossilized groups. Okay, now see the morphological diversities in the Silopsida, Lycopsida, Synopsida, and the Pteropsida support the above view. When you consider the morphological characteristics in the Silopsida, Lycopsida, Synopsida, and the Pteropsida with and matches with these groups with these fossilized forms of the marine algae, you will come to know there is a much similarity between these. Okay, it shows the remarkable morphological features which are matching with each other, and that is why there is a strong proof for the Andrews hypothesis from the fossilized record that these are marine algae, may have given rise to these particular members of the pteridophytes, okay, which belongs to Silopsida, Lycopsida, Synopsida, and the Pteropsida. Okay, now consider the Mehras hypothesis. Okay, the Indian name is there. The Mehras hypothesis. He proposed that the polyphyletic origin of the pteridophytes. Okay, so he is also advocating that the algae is responsible for giving rise to the pteridophytes, and this origin is the polyphyletic. Okay, not from one group, from the many group. Now, see, according to him, both the Bryophytes and the pteridophytes were originated from the hypothetical proto-archaeogoniate group. Okay, what he has hypothesized this time? See, proto-archaeogoniate group, he has hypothesized this group, and from this group, the Bryophytes and pteridophytes were evolved. This is according to the Mehra. Okay, what Mehra's hypothesis is suggesting? It is suggesting that the origin of the pteridophytes is a polyphyletic origin. From the many groups of the algae, there were the creation of the many groups of the pteridophytes, and there is a in between group which is a hypothetical group which is called as the proto-archaeogoniate group. Okay, now see this hypothetical group. Okay, he has imagined this group. Okay, that is why we are calling it as a hypothetical group. Arose earlier from the Chaetophoriaceous ancestor, that is from the green alga. Okay, that is from the chlorophycine members. This group has originated, which is called as the proto-archaeogoniate group. Okay, so chlorophyce algae is there, which has created the proto-archaeogoniate group, and the proto-archaeogoniate group give rise to the Silophytaceous line. Okay, what is the Silopsida? Whatever the Silopsida members are there, this line were created out of this particular proto-archaeogoniate group. Okay, and on the other hand, to the Lycopodaceous line. Okay, so on the one hand, this proto-archaeogoniate group has given rise to the Silopsida, while on the other hand, it has given rise to the Lycopsida. So, Silopsidaceous group is there, line is there in the evolution, and Lycopsidaceous line is also there, which both of these lines were coming out of the proto-archaeogoniate. Okay, so what is Mehra is hypothesizing? He has hypothesized he has imagined a particular group which is called as the proto-archaeogoniate group, and this proto-archaeogoniate group, according to him, is responsible for giving rise to the two lines. The first is Silopsida line and second is the Lycopsida line. Okay, so this is also a hypothesis. He has imagined certain hypothetical group, and from this hypothetical group, the two lines of the pteridophytes were evolved. This is according to the Mehra's hypothesis. Okay, which is also supporting the algal hypothesis. All right. Now consider the second one, that is called as the Bryophytic origin. Okay. Now, as we have studied the algal origin, from your point of view, from your perspective, you can think that there are the many points which are showing the similarity between the algae and the pteridophytes, but also there are many contrasting points as well, okay, which are not discussed by all these hypotheses which are given by the different scientists or even by the algal origin. Okay, algal origin hypothesis is there, which is only discussing the what are the similarities and what are the morphologically comparable characters are there, and what are the anatomical characters which are comparable with the pteridophytes, and on that basis they are saying the algae is responsible for giving rise to the pteridophytes as well as the bryophytes. Okay, now let us consider the second one, that is the Bryophytic origin. Now, of course, while we study the Bryophytic origin, this origin will also give you some morphological characteristics which are very similar between the Bryophyte and the pteridophyte. Okay, now consider many hypotheses have been put forward to support the Bryophytic origin of pteridophytes. Although there is no unanimity regarding the ancestor stock as well as the mode of origin. Okay, so just like the algal hypothesis, the Bryophytic hypothesis also has many authors which are supporting this Bryophytic origin of the pteridophytes, but there is not an unanimity. That literally means there is no one strong agreement between all these scientists. Okay, whatever the authors and the scientists are there which are giving this particular hypothesis, they don't have any common ground to support the Bryophytic origin of the pteridophytes. This literally means that they do not have the clear-cut evidence that the Bryophytes were responsible for giving rise to the Pteridophytes. Now, I intentionally put here the two pictures. In the first picture, here you can see the Sporophytes of the Bryophyte. Okay, so thalloid body is there and the Sporophyte is growing. Food satin capsule, you may have remembered this food satin capsule, and consider this is a Rhynia plant. This is an Pteridophytes. When you observe the first picture and the second picture, you will see the remarkable similarity between the two. Okay, so from this viewpoint, from the morphological viewpoint, we can compare the Bryophyte and Pteridophyte as well, which are showing the remarkable similarities. Observe the Sporophyte of it is a Sporophyte plus the Gametophyte. At the base, you will find the Gametophyte. At apex, what you are saying is the Sporophyte. So it's a combination of Gametophyte and Sporophyte generation in the Bryophytes. Okay. On the other hand, what you are saying here, it's only the Sporophyte. So you can say that the Sporophyte and Gametophyte, this whole structure of the Bryophyte got converted into the one generation of the Pteridophyte because it shows the similar morphological characters. Okay, so but what are the actual evidences to support this Bryophytic origin? There should be some scientific evidence to believe this. Okay, now consider. The base of this hypothesis lies on the similarity between the early vascular plant and the sporophytes of the certain mosses and hornworts. Just we have discussed. If you consider the sporophyte of mosses and the hornworts, this sporophyte and the early vascular plant shows remarkable similarity. Now, when we talk about the early vascular plant, the Rhynia is the prime example. This is the fossilized early vascular plant which is showing the differentiation in their bodies. And it is typically a pteridophyte and not a bryophyte. So early vascular plant like Rhynia, when you compare them with the sporophyte of the bryophyte, you will see the remarkable similarities between the two. And hence, we can say that these sporophytes of the bryophytes may be responsible for giving rise to the pteridophytic, early pteridophytic plants or early vascular plants like Rhynia. Okay, now let us consider which type of these similarities were shown by these particular bryophytes with that of the pteridophytes. See, the characteristics common for both the groups are the heteromorphic life cycle, that is the alternation of generation. As you know, the alternation of generation, that is the heteromorphic life cycle, that is their gametophyte is completely looks different with that of the sporophyte, and this gametophytic generation alternates with the sporophytic generation. So there is a presence of heteromorphic alternation of generation in the pteridophytes. This is also true for the bryophytes, that is, it is showing the heteromorphic alternation of the generation. Okay, heteromorphic alternation of generation present in the bryophytes as well as it is present in the pteridophytes. So this is very similar between the two. Okay, then there are the multicellular sex organ. Of course, as you know, the antheridium and the archegonium, that is the male sex organ and the female sex organ, which are present in the pteridophytes and in the bryophytes. Both are in the both the group. You will find the multicellularity. This is very important. Okay, this multicellularity in the sexual organs of the bryophytes as well as the pteridophytes is taking them very close to each other, because the whatever the antheridium and archegonia which is found in the bryophytes are very similar in the pteridophytes as well. Because you may have remembered drawing the archegonium like this. Okay, these archegonium were similar in the pteridophytes as well as in the bryophytes. Okay, so this similarity, similarity in the multicellular sex organ also taking the bryophytes and pteridophytes very close to each other. Okay, then the motile and the flagellated sperms. Of course, there is always the motile and the flagellated sperms found in the bryophytes as well as in the pteridophytes. So whatever the male gametes are there, they are always flagellated and motile in both the groups, that is in the algae as well as in the pteridophytes. Okay, then the thalloid gametophyte. Okay, thalloid gametophyte were found in the pteridophytes as well as it is found in the bryophytes as well. Observe this. This is a bryophytic thallus and this is a bryophytic gametophyte and this is a pteridophyte gametophyte. It shows the remarkable matching with each other. Their thalloid gametophyte are looks alike, very similar. Okay, and water, of course, is absolutely essential for their fertilization. Bryophytes are dependent for their fertilization on the water. So does the pteridophytes as well. So bryophytes and pteridophytes both require water for their fertilization, which is also showing their similar type of habitat. Okay, why they are like to grow around or in around the water or in the moist area. Okay, so heteromorphic life cycle, multicellular sex organs, motile and flagellated sperms, then the thalloid gametophyte and essentiality of water for the fertilization. All these are the common points between the pteridophytes and the bryophytes, and this makes us to conclude that these groups are very much similar with each other. And when we find the similarity, we can conclude that the bryophytes can be the ancestral stock for these pteridophytes. Okay, for the origin of these pteridophytes. All right. The sixth point, which is very matching between the bryophytes and the pteridophyte, is the retention of the zygote within the female sex organ and the embryo development from it. This is very common between the bryophytes and the pteridophytes, where there is archegonium, and in the archegonium what we have is the egg, and it's the non-motile egg. Okay, and when the sperm or the female gametophyte or the male gametophyte reaches here, and there is a fusion and there is a formation of zygote, this zygote is retained in the female sex organ, that is the archegonium. Okay, that means the archegonium is retaining the zygote in the pteridophytes as well as in the bryophytes. So bryophyte, whether it is a pteridophyte or bryophytes, in both the archegonium, that is the female sex organ, is responsible for retaining the zygote. Okay, and this makes them very similar with each other. Now also see, the plants and the spore surfaces are covered with the cuticle. Whether it's the bryophytes or the pteridophytes, their plant surfaces or their spore surfaces are covered with the cuticle, which is made up of the cutin. Okay, which checks the loss of water during the transpiration. Okay, so the cuticle is also making them very similar with each other. In that sense, the bryophyte and pteridophyte both are possessing this particular type of the cuticle in their bodies. So from all these 7 points, you came to know that the bryophyte and pteridophyte shows the remarkable morphological as well as the anatomical features, which makes them very close on the evolutionary line. Hence, we can support that these pteridophytes may have originated from the bryophytes. Okay, now consider Antherocity theory. Okay, now this is an important theory while we are describing the bryophytic origin of the pteridophyte. This was advocated by the Campbell. Okay, now see, according to him, this theory, the sporophyte of the Anthoceros shows many characteristics comparable with that of the sporophytes of early vascular plants. Okay, for example, if you consider the early vascular plant like Rhynia and its sporophyte and its sporophytic plant body, as you already know, the sporophytic plant body of the Rhynia is like that. Okay, we have seen in the last picture as well. This type of this sporophytic plant body is shown by the Rhynia, and this sporophytic plant body of the Rhynia shows the comparable characteristics with the sporophyte of Anthoceros. Okay, so whatever the sporophyte of Anthoceros is there, it's showing the remarkable matching with the sporophytes of the early vascular plant. Okay, when we talk about the Anthocerotian theory, okay, Anthocerotian theory, what it says, the sporophyte of Anthoceros and the sporophyte of early vascular plant, whatever the early vascular plants were there, the sporophyte of these early vascular plants, for example Rhynia, matches typically with the sporophyte of the Anthoceros. Okay, now consider the matching points. The sporophytes bear the rhizoids instead of the roots. Okay, the sporophytes of the early pteridophytes shows the presence of rhizoid instead of roots, and this is also true for the sporophyte of the Anthoceros. Then the mechanism of the indefinite growth through the meristematic tissue is present in both, that is in the Rhynia-like plants and in the sporophyte of the these are Anthoceros. Okay, so mechanism of the indefinite growth is present in them, and presence of columella, that is in the Horny Phyton and as in the Anthoceros, makes them very similar on the anatomical ground. Okay, so Horny Phyton, as you already know, we have discussed it, it's an extant connecting link between the bryophytes and the pteridophytes. Okay, so what are the similar points? The sporophyte bears the rhizoid instead of roots, plus they are showing the indefinite growth through their meristematic tissues, and they shows the presence of columella. Okay, so we are talking about the Antherocity theory. Now, according to the Antherocity theory, the sporophyte of Anthoceros and the sporophytes of the early vascular plant shows a remarkable similarity between them. And these are 3 points correspond to this, which are saying the sporophyte bears the rhizoid instead of roots, and they both are showing the indefinite growth through the meristematic tissue, and they both shows the presence of columella. Okay, now consider, there is a limited tissue in the spore production. Okay, in both the cases, whether it's the Rhynia or the sporophyte of the Anthoceros, in both the cases they are showing the limited presence of the tissue for the spore production. As you already know, we have discussed the sporophytic evolution in the bryophytes. In these, the sterility is there and the limited tissue is available for the production of the spores. This is also true for the Rhynia. Okay, now see, the capsule comprises the photosynthetic tissue and provided with the stomata. Of course, whatever the capsule are there, they are showing the presence of the photosynthetic tissue, plus there is a presence of the stomata, and this makes them very similar with that of the sporophyte of Rhynia or the early vascular plant. Thus, the early vascular plants like Rhynia were derived directly from the Anthoceros like ancestor. Okay, so according to the Antherocity theory, there is a sporophyte of the Anthoceros, and there is a sporophyte of the early vascular plant like Rhynia, and they are showing remarkable matching with each other. Okay, what they are showing, they are showing morphological anatomical features alike, and that is why this theory is saying that the Anthoceros like ancestors were there to create the Pteridophytes out of the Bryophytes. Okay, now consider. See, Smith, this author is strongly advocating the Anthocerotian hypothesis, that is the Pteridophytes were originated from the Bryophytes, but with some modification. Okay, Smith is advocating the Bryophytic origin, but he has given some modification. He postulated that the Anthocerotian sporophyte had shifted the meristematic region from base to the apex. This is very important. As you already know, the Anthoceros is a typical Bryophyte, and as you know, the main generation in the Bryophytes is the Gametophyte, and Gametophyte is like this. Observe in this picture, this is the Gametophyte. Okay, this is the Gametophyte of the Anthoceros. And according to the Smith, whatever the growing points are here on this gametophyte, it has shifted from base to the apex. This growing point has shifted from base to the apex, and that is why there is a formation of aerial shoots in the Pteridophytes. Okay, so at this its growing point got shifted from base to the apex. There comes the erect habitat, and this erect habitat later gave rise to the Rhynia like early vascular plants on the land. Okay, so due to the shifting of the meristematic region right from the base to the apex, there is a formation of the Pteridophytes out of the Bryophytes. This is according to the Smith. Okay, thus the initiation of the dichotomy was established in the apical meristem, and the spore formation become restricted to the branch apices. Okay, this is very important. The whatever the apical meristems is there, it initiated the apical dichotomy. That is, there is a separation of the branches. There is a dichotomous branches were created in a aerial way. Okay, in the erect fashion, and the spore formation has become restricted to the branch spices only, because the branches contain the apical growing point, and that is why the spore formation structures were restricted to the apices only. This is according to the Smith. So Smith is also advocating the Bryophytic origin for the Pteridophytes, but with a certain modifications. Okay, he has given some own points. Okay, just for example, the one point is there, one prominent point is there. What he is saying, according to the Smith, the apical, the meristematic region, because you must have cleared about this meristematic region. Meristematic region are those regions where there is a rapid multiplication of the cells. Okay, rapid multiplication of the cell is there, and due to this rapid multiplication, there is a growth and development of the gametophyte or the sporophyte. Okay, wherever there is a meristematic region, it is responsible for the growth. Okay, so according to the Smith, what this meristematic region were transferred from the base to the apex, which literally give rise to the early vascular plants in the Pteridophytes. Okay, now see, the columella, whatever the columella were present there, eventually evolved from the vasculature of the land plant. Okay, to form the vasculature of the land plant. Okay, so what he is saying, the columella structure which is present in the Anthoceros, it has converted into vasculature, that is the vascular bundle, xylem and phloem. The xylem and phloem were created out of the columella of the land plant, for example, the Rhynia. So in the course of the evolution, the sporogenetes, okay, the sporogenetes evolved. The sporogenetes were the group of the plants which literally showing the characteristics of Bryophytes as well as the Sporophytes. Okay, now see, the sporogenetes consist of the merry parallel oriented sporangial stalk that terminates the elongate capsule containing the central columella. Okay, sporogenites, the sporogenite, it consists of a many parallel oriented sporangial stalk. Okay, like this many parallel sporangial stalks were there that terminate into the elongate capsule. Okay, like this, it terminates into elongate capsule which contains the central columella. Okay, so these sporangionates were responsible for later giving rise to the early vascular plants. That is, this sporogenite is strongly showing the Bryophytic characters, and these Bryophytic characters later got created into the Pteridophytic characteristics. Okay, so these are sporogenetes. Whatever the sporogenetes were there earlier, these sporogenetes were responsible for creating the Pteridophytes later in the evolution by shifting the meristematic region from base to the apex. Okay, from base to apex, the meristematic region has shifted, and from the columella has got converted into the vasculature that is in the xylem and phloem. So this is how, according to the Smith, the evolution from the Bryophyte to Pteridophyte has occurred. Okay, now consider. Later, the Horny Phyton, which is the missing link between the Bryophyte and Pteridophyte, evolved from the sporogenetes through the distal dichotomy. Okay, so these are Horny Phyton, which is the extinct group now. Now this has been evolved. this Rhynia plant, this has been evolved from the distal dichotomy. Observe here. Here you will find the distal dichotomy is there, and this due to this distal dichotomy, this Horny Phyton got created out of the Bryophytes. Okay, the lower parenchymatous corm and the columellate capsule of the Horny Phyton are interpreted as a transitional form between the Anthoceros and Rhynia type of vascular plant. See what the Horny Phyton is showing. It is showing the parenchymatous corm. It's the anatomical structure which is present in these Horny Phyton. Okay, so parenchymatous corm are there and columellate capsule is there. These two structures were considered as the intermediate structure or the transitional structures between the Anthoceros and the Rhynia type of vascular plants. Okay, so Anthoceros on the one hand possesses the typical type of the capsule, while on the other hand the Rhynia is showing the presence of the corm. So these intermediate structure, columellate capsule and the parenchymatous corm, considered as a transitional structure between the Anthoceros and the Rhynia. Okay, which are transiting right from the Anthoceros to the Rhynia. Okay, the further elaboration of the basal part of the Horny Phyton eventually give rise to the rhizomatous stem of the Rhynia. So whatever the basal portion is there in the Horny Phyton, it eventually give rise to the rhizomatous stem. Okay, like this rhizome like stem with the rhizome is created out of the basal portion. Okay, so the on the evolutionary line, what is this particular Smith theory is saying, which is advocating the bryophytic origin, that how the Anthoceros type of the morphological plant got converted into the morphological features of the Rhynia-like plant. And for this, to prove this, he has given the example for the shifting of the meristematic cells, and then there is a conversion of the columella into the xylem and phloem like structure, and the basal portion into the rhizome like stem of the Rhynia. Okay, so the typical morphological characters which are present in the Anthoceros got converted into the special structure which are present in the Rhynia over a period of time through the course of the evolution. This is according to the Smith. Okay, now consider the Stobular theory. Okay, now see, this Stobular theory was proposed by the Bower. Okay, Bower has given this particular Stobular theory, and as you know, Bower is a famous bryologist. See, according to this, the Pteridophytes have evolved from the Anthoceros type of ancestor. That means the Stobular theory and the Bower is actually supporting the theory of the bryophytic origin for the Pteridophytes. Okay, he is also saying that the Anthoceros type of ancestors were there for the Pteridophytes. He suggested that the well differentiated Sporophyte evolved by the progressive sterilization of a potentially sporogenous tissue. We have already discussed that during the sporophytic evolution in the Bryophytes, that there is a progressive sterilization in the potentially sporogenous tissue. So he is also saying the same thing, that the Anthoceros, whatever the Anthoceros plants are there, through these plants there is a progressive sterilization in their sporogenous tissue, which later got converted into the Pteridophytes. Okay, so this is the simple idea which is put forward by the Bower, that he calling it as the Stobular theory, and according to this theory, in the Anthoceros there is a progressive sterilization of potentially sporogenous tissue, which ultimately leads to the formation of the Pteridophytes. Right? See, according to this theory, here is the one example, that is the Lycopodium selago, which is a Pteridophyte. Okay, in which the sporophylls are arranged singly throughout the body surface without organizing into the definite stabuli, arose from the Anthoceros ancestor through the following steps. See, this is a plant, it is called as the Lycopodium selago. And what is the specialty of this Lycopodium selago, which is a pteridophyte? That it possesses the sporophylls which are arranged singly throughout the plant body surface. Okay, throughout its plant body surface, it is showing the presence of the sporophylls throughout the plant body. Okay, not just the one part, but throughout the plant body it is showing the presence of these sporophylls. And according to the Bower, this Anthoceros ancestor has created this particular type of the Lycopodium selago plants. Okay, by the following step, he actually given the events which leads to the formation of Lycopodium selago like plants from the Anthoceros. Now consider, there is a formation of the continuous sporogenous tissue just outside the central columella in the Anthoceros. Okay, in the Anthoceros, when we talk about the central columella region, there is a continuous formation of the sporogenous tissue just outside to this central columella. Okay, so anatomically there is a presence of the central columella in the Anthoceros, and from this central columella, just outside of this central columella, there were the formation of this sporogenous tissue, continuous formation of sporogenous tissue. Now this this sporogenous tissue becomes superficial. Okay, what these points are showing? These points are showing steps in which these particular Anthoceros got converted into the Lycopodium like plant. See, in the first step, what happened? What may have happened? according to the Bower, that the sporogenous tissue were continuously formed outside of the central columella. After that, this sporogenous tissue once formed outside the central columella, it becomes superficial. How it become? It becomes superficial, and this sporogenous tissue was segmented into the separate masses of the intercalated sterile tissue. That is, this sporogenous tissue, this got segmented. The segments were formed in this particular tissue, and later it got converted into the it got separate masses by an intercalated sterile tissue. So whatever the sterile tissue are there, this sterile tissue got separates this particular sporogenous tissue. So the sporogenous tissue, which is formed in the first step, which got superficial in the second step, in the third step they got separated from each other by the intercalated sterile tissue. So whatever the mass of the sporogenous tissue were formed, this sporogenous mass of the tissue got separated from the one another by the presence of the intercalated sterile tissue. Now this has been evidenced in some members of the Antherocity. Okay, if you study the Antherocity members, you will find such a type of differentiation has already occurred in these particular kinds of the plants. Okay, now consider the isolated and the segmented sporogenous mass become more superficial, and thus alternating masses of a sporogenous tissue and the green sterile tissue were formed. Okay, so this is how the Lycopodium selago got created. What is the first step? See, consider this is the central columella in the Anthoceros, and in the central columella there is a outside of this central columella there is a formation of the sporogenous tissue. Now this occurred in a first step. Okay, now in the second step, what happened? This becomes superficial like this. Okay, it becomes superficial like this, and in the third step, what happened? The mass of a sterile tissue formed in between these sporogenous tissue, so they got segmented. Okay, just like this. So segmentation occurred in the sporogenous tissue, and this the feature which is creating the segment is the sterile tissue. Okay, now later these sterile tissue were become green. Okay, and the sporogenous tissue remain as it is. So there is a segments of the sporogenous tissue which is alternating with the green tissue. Okay, so thus the alternating masses of the sporogenous tissue and the green sterile tissue were formed like this. Okay, see, the green tissue expanded laterally to form the leaf. Later this green tissue which is coming out of from the sterile tissue, it became leaf, like see in this plant it became leaves. Okay, and thus the sporogenous tissue, that is the sporangium, come adaxially to the base of the leaves, that is the Lycopodium selago. So whatever the sporogenous tissue was present in between these green tissue, it came to the base of the leaves. Okay, and it becomes sporangium. Okay, like this. So it ultimately become the Lycopodium selago, because Lycopodium selago constantly is showing the sporophylls, and at the base of the sporophylls there is the reproductive structure. So this is how the particular Anthoceros type of the plant got converted right from the anatomical feature to the morphological feature into the Lycopodium selago. So what Bower is saying? Bower is saying the anatomical and the morphological features of the Anthoceros over a period of time, over a few steps, by converting slowly by steps, got converted into the pteridophytic plant, for example Lycopodium selago. Okay, so this is according to the Bower. All right, now consider the Phyton theory. Okay, now this Phyton theory was given by the Seleucus. Okay, Seleucus was a scientist who has given this particular Phyton theory, and according to this theory, the fundamental part of the sporophyte was a leaf. Okay, what he is saying, the fundamental part of the sporophyte was leaf and not the axis or stem, and axis formed later by the fusion of the leaf bases. Okay, so he has made some different contrasting points regarding the origin of the pteridophyte from the bryophyte. What he is saying, the fundamental part of the sporophyte was the leaf. Okay, which structure were first formed out of the bryophytes to become pteridophyte is the leaf, and after the formation of leaf, through the fusion of these leaves, where axis and stems were got created. This is according to the Phyton theory, who is saying the leaf is formed first in the evolution from the bryophyte to pteridophyte, and from the fusion of these leaves there is a formation of the axis or the stem when the leaf bases fuse with each other. That is, earlier the leaves were formed like this. Okay, leaves were formed like this, and these leaves, after the fusion of their bases, like this, fusion of two bases. Okay, this has created the stem or the axis in the pteridophytes. Okay, this is according to the Seleucus guy. Okay, which has given the Phyton theory. Now see the protocorn theory. Okay, which is also advocating that the pteridophytes were evolved from the bryophytes. This has been given by the tube. Okay, now what he's saying, consider this protocorm, this structure is anatomical structure. Okay, it's an anatomical structure which is found in some of the plants. Okay, now consider this. These according to this theory, the sporophyte of the primitive pteridophyte was an undifferentiated mass very much like of the gametophyte. As you already know, the gametophyte where the looks like an undifferentiated mass of cells like this, and according to the tube there is an undifferentiated mass of the sporophyte as well in the earlier plants. Okay, this has been substituted by the occurrence of the protocorm in some species of the lycopodium. Like lycopodium is showing the anatomical characteristic which is called as the protocorm, and this protocorm is the earlier structure, the ancient structure according to the tube, and this ancient structure were earlier present there, and after the differentiation in this protocorm structure, there is a formation of the pteridophytes. Okay, so these are protocorm structure according to the tube is coming from the bryophytes to the pteridophytes, and later this particular protocorm structure is responsible for giving rise to the modern day sporophyte of the pteridophytes. Okay, see the undifferentiated mass very much like the gametophyte. This has been substituted by the occurrence of the protocorm in some species of the lycopodium. That is, he is actually saying that the sporophyte of the primitive pteridophyte, how it was like? It was like the undifferentiated mass, which is called as the protocorm. Okay, nowadays protocorm is already found in the lycopodium, so he is considering the anatomical feature called like protocorm as the connecting link between the bryophytes and the pteridophytes. Okay, so to consider the connecting link anatomically between the bryophyte and pteridophyte, the tube has used the protocorm as the structure which connects the bryophyte with that of the pteridophyte. Okay, but this is Bower has strongly disregarded this protocorm theory. Okay, now why he disregarded? Because what he is saying, he concluded that the protocorm is merely the adaptive feature to the certain special environmental condition. That is, whenever the lycopodium, you can observe in this picture, these are the lycopodium species. The lycopodium create this protocorm as an anatomical structure is an adaptive structure to certain environmental condition. Okay, so if the lycopodium species has face certain kinds of environmental stresses in the in and around its occurrence, and that is why it has created an adaptive structure which is called protocorm, and it has nothing to do with the evolution. This is according to the Bower. Okay, so on the one hand, the tube who is strongly connecting the protocorm structure with the evolution, on the other hand, the Bower disregarded this protocorm structure as merely an adaptive structure. Okay, for the lycopodium species only, and it has nothing to do with the origin of the pteridophytes. Okay, so these are the contrasting views between the two scientists, that is the tube and the Bower. Okay, now let us consider what are the modern studies suggesting about it. Okay, all these views, algal views, the pteridophytean views, or the bryophytean origin views, all these scientists are saying they from their own point of view, and majority of them are the hypothesis, and majority of the time they have hypothesized some imaginary group as well. So is there any definite evidence to say something about the origin of the pteridophytes, whether it is the algal origin or the pteridophytic origin? But whether it's algal origin or the bryophytic origin, but there should be the strong proof that the which support whether it's the algal or the bryophytic origin. Consider the modern studies give some focus on this. See the modern studies of the cell ultrastructure, then the biochemical nature and the molecular studies like the rRNA sequence, then the molecular sequence data from the plastids, nuclear and the mitochondrial encoded genes. This is very important because the plastid like chloroplast contains the DNA, nucleus contains the DNA, mitochondria contains the DNA, and when we taken out this DNA and sequence this DNA for which type of genes it's encoding, it suggests that the bryophytes are not the ancestors of the vascular plant. So here comes the strong evidence. According to the molecular studies, when we study the DNA of the bryophyte, pteridophytes and the algae, it has a strong proof that the bryophytes were not the ancestors of the vascular plants, because on the molecular basis they do not match with each other. Okay, so what modern studies are saying? Modern studies are saying that the bryophytes were not the ancestral stock for the vascular plants like a pteridophytes. So from where these vascular plants came? See, it has been indicated that both the bryophytes and pteridophytes have been evolved from the green algal ancestor, probably from the coleochaetes closely related to the chlorophytes, that is the green algae, and the mosses appears to be the sister group of the Trachyophytes. So finally, we have proved that the algal origin of the bryophytes and pteridophytes were strongly supported by the modern studies. That is, the biochemical studies, molecular studies, ultrastructure studies were there. And from these studies, we can clearly say that the bryophytes were not the ancestors for the vascular plants, but the algal group. Actually, the green algal ancestors are there from which the bryophytes and the pteridophytes have got evolved. Okay, so this is very strong statement. Okay, they are saying probably from the coleochaetes, that is which is the group of chlorophyceae, okay, which is closely related to the chlorophytes, and the mosses appears to be the sister group of the tracheophytes. Okay, so from these molecular studies, from these particular ultrastructure studies, from these biochemical studies, we conclude that the bryophytes were not the actually the ancestral plants for the pteridophytes, but the green algas are responsible for giving rise to the bryophytes as well as the pteridophytes. Okay, so further studies are also going on this particular topic. So with time we will know more about these particular subjects. Okay, now consider thus the coleochaetes is an excellent model for the algal ancestry of the land plants and may be a modern representative of the algal group that give rise to the land plants. Okay, so in the algal group, what we have is the coleochaetes, and these are coleochaetes is an excellent model when we study them from the molecular basis. And from this particular coleochaetes group, the bryophytes, whether it's the bryophytes or the pteridophytes, have got evolved. Okay, so molecular evidence is strongly suggesting that the bryophytes and the pteridophytes, both plants, are created out of the coleochaetes type of plants, which belongs to the green alga. Okay, now see the evidences in this support. Okay, now if we consider the evidences or molecular evidences, see the coleochaetes is the soil alga, of course, it in the order coleochaetales and the subfamily of the chlorophyceae. Okay, which produces the small vegetative thallus as the major and the haploid part of its life cycle. Okay, this particular alga which belongs to the coleochaetales, see, it produces the vegetative thallus, which is the dominant phase of its life cycle. Okay, because as you already know, in the algae the haploid is a dominant phase in their life cycle. Okay, so it is producing the vegetative thallus, which is an haploid thallus, which form the major part of its life cycle. Now consider this thallus produces the egg cells towards the periphery. Okay, what is it is producing? It is producing the egg cell towards its periphery, just like the bryophytes and pteridophytes. Just in the pteridophytes as well, the gametophyte is producing the egg cells in their gametophytes. Right now, once the egg is fertilized, the zygote is retained in this particular portion. This is very similar with that of the bryophytes and pteridophytes, because zygote, whenever the egg is got fertilized and the zygote is got created, this zygote is retained, on the body of the female gametophyte or inside the female sex organ on the parent thallus, right, and supplies with the nutrient through the specialized transfer cells. Okay, and it has taken all the nutrition from the parent thallus, and this characteristics makes them very similar with that of the bryophyte and pteridophyte. Okay, then this zygote undergoes the cell division, while it still retained on the parent plant. This is also similar with the bryophytes and pteridophytes, that the zygote, whatever the zygote is created there, this zygote, it undergoes the cell division while it is still on the parent gametophyte, right? And there is a greater number of meiotic divisions as well, meiotic products as well, because 8 to 38 cells were formed through the meiotic processes. Okay, so this all makes this specialized algae very similar with that of the bryophytes and the pteridophytes. Okay, and that is why it is clearly indicating that the bryophytes and the pteridophytes are originated from the algal ancestry. Okay, so finally we have the clue about the origin of the pteridophytes, that the pteridophytes were originated from the algal stock and not from the bryophytic stock. All right, now consider the reproduction with the specialized male gametes similar to the male gametes in bryophytes. Okay, bryophytes we showing the male gamete. The similar male gametes were also shown by these algae. Then the phragmoblast, which is formed during the cell division pattern like those of the land plants. In the land plants, there is also the formation of the phragmoblast, and these phragmoblasts were also created in these algae as well. Now see the sporopollenin in their inner wall of zygote. So zygote of these particular algae showing the presence of sporopollenin, and this sporopollenin chemical compound is also present in the spores of the pteridophytes and the pteridophytes, right? So these pteridophytes also contains the sporopollenin in their spore structure, while the sporopollenin is also present in the zygote of these algae. Then the presence of lignin, a principal component of the land plant. Okay, lignin is also present in these algal groups, and these lignin were also found in the land plants as well. So the chemical similarities were also there. Then the group 2 types of the introns. Okay, this is the molecular study. As you already know, the genome is divided into exons and the introns. So the group 2 types of the introns in the chloroplast of the genome, like in the land plants. Okay, just like the land plants which are showing the group 2 type of the intron, it is also found in the chloroplast genome of the algae. Okay, so these are all molecular studies and the biochemical studies or the morphological studies showing that the algae are the ancestral stock for the bryophytes as well as for the pteridophytes. Okay, so this is all about the origin of the bryophytes. Okay. So this is all about this topic. Okay, thank you very much for watching this video. Please like this video, share this video with your friends who are studying the botany, and if you haven't subscribed to the Botany channel yet, please subscribe to the Botany channel for UPSC examination. Again, thank you very much for giving your time. See you in the next video lecture. Thank you.